So, hello students, welcome to Junior Science Olympiad. This is Basavaraj, sir, your biology master teacher. Now, students, in the previous class, from this particular chapter, basic to advanced, we covered all the basics of the chapter, right? All of you might remember, students, this was the first class. In this 15 minutes of class, we finished most of the basics which you need to know to understand this particular class is already covered here. So, I would recommend all the students, this is a small recommendation to all the students to quickly go and watch this lecture first before watching this lecture, because if you want to really understand what is a light reaction, you need to know the basics, right? So in the previous class we started off with what exactly is photosynthesis, then we covered what exactly is the meaning of pigment, then we also covered where exactly the entire process of photosynthesis takes place. For example, here if you look at, this is a structure of chloroplast. This is a structure of chloroplast, and the entire process of photosynthesis takes place right here, right? All of you might be aware it is a double membrane structure, outer inner membrane. Then it is clearly seen here, students, clearly seen here, right? There is coin-like structure called as thylakoid. Entire thing is called as a grana here. Then we have stromal lamellae here. Done. Now, students, if you clearly see here, on top of thylakoid, we have PS1, PS2, both are present on the thylakoid here. On the stromal lamellae, we have only PS1. So only PS1 is present here. So this is a very crucial information if you want to understand the light reaction. This is the basics for light reaction, students. Here, I'll repeat again, students. Both PS1 and PS2, not PlayStation. I'll tell you what is PS1 and PS2. Don't worry. Both PS1 and PS2, both are present here. And on the stromal lamellae, we have only and only what? We have only and only PS1 here. PS1. This much all of you know. This was discussed in the previous class also. Right? Previous class also. Then we learned about pigments. Then we learned about this experiment right here. Then we learned about different types of pigments, right? We learned there is something called a chief pigment. Chief pigment is like your school principal, highest power, everything. And there is something called as auxiliary pigments, chlorophyll B, carotenoids, xanthophyll. They are like the teachers. They are like the teachers who has to report to chlorophyll A, right? So here what happens here is that, if you remember, I told you in the previous class, every single time you are making some noise in the class, your teacher will say, first of all, it is a fish market, yes or no? Then the teacher will say, I want pin drop silence. Then even if there is still noise in the class, teacher will be like, I will send it to principal, principal give you TC. So teacher always sends the student to principal's room to give the TC, because the authority, the ability to give TC is for principal. Similarly, students, here chlorophyll B, carotenoids, xanthophyll, they cannot do any type of reaction. They cannot do reaction because they do not have the enzymes for reaction. They do not have the enzymes for reaction. All the reaction, like how TC is given, all the reaction can be given by chlorophyll A. So what will they do, students? This chlorophyll B, carotenoids, xanthophyll, they will trap the light. They will trap the light energy and they will transfer the light energy to chlorophyll A. Transfer the light energy into chlorophyll A, because all the work, all the enzyme action, chlorophyll A does it. Also, it's called as chief pigment. Okay. Now, this was discussed in previous class also. I told you all, students, because of this entire arrangement, right, the wavelength of light which plant can absorb increases. See, there is generally a wavelength of light called as PAR, photosynthetically active radiation. Photosynthetically active radiation. It is from your 400 nanometer to 700 nanometer. Okay. Sometimes even some textbooks say even 300, but will 400 700 nanometer. Now, in this wavelength of light, right, chlorophyll A will only work at 680 nanometer. Now, there is one more chlorophyll A that works at 700 also, but let's say 680 right now. 680 to 700 nanometer. Now, what about the remaining wavelength of light? 400 to 500, 500 to 600, 600 to 650. What about those wavelength? So, in those wavelength, right, let's say 500 to 550, right? 500 to 550, 550 to 600, 600 to 650. So in these wavelength of light, students, in these wavelength of light, who is capturing this light? For example, let's say here we have carotenoids, here we have xanthophyll, then we have chlorophyll B here, students, chlorophyll B. So they can absorb this light of this wavelength and transfer it to chlorophyll A, the energy. Similarly, students, how a principal cannot control the entire school. A principal cannot go to every single classroom and teach. Is it possible? No, not possible. So what does the principal do, students? Principal will make sure all teachers are present in every single classroom and the teachers will report to the principal. Here also, this chlorophyll A cannot be present in all the wavelength. It cannot capture all the wavelength, not possible. Right? Similarly, it has multiple teachers who can go to every single classroom, record the wavelength, report to the chlorophyll A. Now you tell me, students, what will happen if the principal has to go every single day to every single class, every single period to teach? The principal will go mental, like not possible. Similarly here also, students, if this chlorophyll A, if it is to observe all the wavelength of light from 400 to 700, if everything is absorbed by chlorophyll A, it will also become mental. The meaning of mental here is it undergoes photooxidation. So we do not want chlorophyll A to go photooxidation. So that is why we have the teachers. Who are the teachers here? Teachers are going to be the auxiliary pigments here, students. So I hope that concept is clear to all of you. What is the main function of chief pigment and auxiliary pigment? Now, students. Now we come to a small topic here, very small topic called as action spectrum as well as absorption spectrum. It's very easy. Listen to me just now. Listen to me. Okay. Students, first of all, if you not like the video, quickly like the video also right now, because it is very important to like the video, because when you like it, the reach of this video increases and obviously many students will be able to look at the lecture. Very simple algorithm, right? Now, what exactly is absorption spectrum? Now, all of you understood, right? All of you understood there are different types of pigments here. We have chlorophyll A, chlorophyll B, carotenoids, xanthophyll. Now, I also told you every single pigment will absorb different wavelength of light. Here, every single pigment will absorb different wavelength of light. That is what is absorption spectrum. Students, that is what is your entire absorption spectrum. Students, entire absorption spectrum here. Okay. Absorption spectrum. Okay, entire absorption spectrum, students, entire absorption spectrum tells you that which wavelength is absorbed by which pigment. That's all. That's all, students. Can all see the board? Is visible, right? Right? So, absorption spectrum tells you that which wavelength is absorbed by which particular pigment here. Okay, right? Okay. So, if we can see here, you can tell here, students, chlorophyll A is getting absorbed here and here, both the places. Both the place. Chlorophyll is getting absorbed here. Chlorophyll B is getting absorbed here. Carotenoid is getting absorbed here. So every single pigment gets absorbed at a particular wavelength. Okay, see here. Pigments have the ability to absorb light at specific wavelength. The absorption spectrum of leaf pigment indicates that wavelength of light. Which wavelength of light? Right? Wavelength of light absorbed by each pigment. Right? So we can see here, students, in much more simpler words right now. Here. See here. Where did that go? 1 second, students. Here. Which wavelength is absorbed by which pigment at a particular wavelength? That's all. Very simple it is. Very simple. So let's say at 400 wavelength, chlorophyll is getting absorbed. Chlorophyll is absorbing the light, basically. Chlorophyll is absorbing the light there. So every single wavelength has a fixed wavelength which will be absorbed light. That is called absorption spectrum. This spectrum shows wherever pigment is getting absorbed. Okay. Now, students, there is something called as action spectrum. Okay. Now, what do we mean by action here? The meaning of action here is actually photosynthesis. Photosynthesis. So, this wavelength, here we have wavelength, here we have rate of effectiveness. So this particular action spectrum tells you at which wavelength, photosynthesis is happening. That's all. If you can see, students, at this point here and this point here, at these two regions, photosynthesis is happening more. That's all. That is called as action spectrum. It is a spectrum which is showing at which wavelength the entire process of photosynthesis is happening. That's all. Okay. That's all, students. See, action spectrum shows the effectiveness of different wavelengths of light for a particular reaction. Shows which wavelength of light is most effective utilized in a specific chemical reaction. So here this is for chlorophyll A. If you can see here, students, one more diagram I'll show you. See here, students. See. Let's compare now. Let's compare here, students. Here we have chlorophyll B is getting absorbed here, right? Now chlorophyll cation is getting absorbed here, right? Cation is getting absorbed here. Now can they do photosynthesis? No, they cannot do photosynthesis. What they can do, these carotenoids and chlorophyll B, they can take the energy and give it to the main boss, a main principal. Who is the main principal here? Chlorophyll A. Can you see, students? This light blue color, chlorophyll A. Now, if you can notice very clearly here, students, chlorophyll A is getting absorbed here and it is getting absorbed here. Now, in these two regions, do we see a peak again here and here? Yes, we see a peak here and here. What does it tell you, students? At this peak here and this peak here, in these two peaks, what is happening? Light absorption is happening. Along with that, photosynthesis is happening. Photosynthesis is happening. So if you can overlap them, students, you will get a somewhat this structure. Wherever there is absorption, wherever there is chlorophyll A absorption, we can see photosynthesis happening at the same peak. So this is your absorption spectrum. Absorption spectrum is showing you at which wavelength light is being absorbed by chlorophyll A. And this is your action spectrum. It is telling you at this wavelength and this wavelength photosynthesis is happening. So action spectrum and absorption spectrum. Now here is a very what do I say, advanced level concept. Listen to me. Very simple advanced level concept now. Now students, you can see here between 500 to 600 there is no chlorophyll A absorption. Not at all. Chlorophyll is not getting absorbed at all here. If chlorophyll A is not getting absorbed here, how do we see this? Still there is more photosynthesis still happening. This black line should have come here, no? This black line should have come here. So even though there is chlorophyll A is not getting absorbed here, why is there still some amount of photosynthesis still happening? Why, students? Because answer is very simple. Students, the answer here is the auxiliary pigments. So even though chlorophyll A is not getting absorbed, at 500 to 600 nanometer, not getting absorbed, but other pigments are getting absorbed. So when those pigment absorb the light, but not pigments are not getting absorbed, those wavelengths of light are getting absorbed by these pigments, right? These pigments, chlorophyll B, carotenoids, xanthophyll, they are absorbing the light and then giving the energy to chlorophyll A. So that is, student, that is why, because of all the different types of auxiliary pigments, even though there is no chlorophyll A doing any work here, okay, no chlorophyll A is not absorbing any light, here. It is not absorbing any light here, right? The other auxiliary pigments are absorbing the light and giving the energy to chlorophyll A, and chlorophyll A is doing some work here. So chlorophyll A will work, because even though, right, teachers are absent, principal has to work. Okay, principal has to work. Okay, so principal is working here, but even though principal is working here, student, principal is working here, right? Chlorophyll is working here, but it is not absorbing any light. Here. Okay, now students. Now next concept comes. What is LHC? LHC is called as light harvesting complex. So the main function of LHC is to absorb the solar radiation. Okay. Now, what is LHC made up of? LHC is made up of antenna and reaction center. Now, what is reaction center? Reaction center is nothing but a chief pigment chlorophyll A. What is antennae, students? Antennae are the axillary pigments. So these axillary pigments and reaction center together are called as LHC, right? Light harvesting complex. The main function is to harvest the light. The main function, students, is to harvest the light here. Then, right, the pigments are organized into two discrete photochemical lights harvesting complexes within the photosystem. LHC are made up of hundreds of pigment molecules bound to the protein. Okay, so we have LHC here now, students. LHC is here. Now, that brings us to the next concept called as photosystem. Photosystem. Now, students, let me just draw a diagram for you to understand the difference here. Okay, let's say this is a photosystem right here. Okay, photosystem right here, students. Now, what is one photosystem made up of? One photosystem is actually made up of three parts. We have LHC here. Yes, LHC is part of photosystem here. LHC is here. Then there are certain electron carriers here. Electron carriers. Then we also have certain enzymes. Enzymes plus even proteins here. Proteins. Enzymes are also proteins anyway, but right, not all. But so proteins and plus enzymes are here. Electron carriers and LHC. That is what makes up the photosystem. Photosystem. Now, students, this all of you now know what is a LHC. All of you know what is a photosystem right now. The next step is to understand there are two types of photosystems here. There is photosystem 1 and photosystem 2. Now again, students, you will realize that here photosystem 1 is at P700, photosystem 1 is at P700 and photosystem 2 is at 680. You're like, why, sir? One should always have lower number, right? You technically, students, PS1 is supposed to be 680, then we get 700, right? In numerical order. Then why have we given P700 as 1 and photosystem 2 as 680? Because, students, it is based on order of discovery. Photosystem 1, this was first discovered, that is why this photosystem 1 was called. And this photosystem 2 was discovered later on, so it is called as P680. Okay, order of discovery. Now, students, what is this P? P is basically photosystem. What is the number next to it? Number is the wavelength of light it can absorb and work at. Okay, so P1, photosystem 1 can work at 700 nanometer, photosystem 2 can work at 680 nanometer. So those are the wavelengths of light where it can actually work and absorb the light. Difference you understand, students. It can absorb the light of wavelength of 700 nanometer. It can absorb the light of the wavelength of 680 nanometer. So 700, 600. PS1 lies on the outer membrane of the thylakoid, outer surface of thylakoid here. PS2 lies on the inner surface of thylakoid. This is very important. Inner surface of thylakoid. Okay. Now this all of you already know. It is found in both grana and stromal lamellae. What? PS1. While PS2 is only found in the grana lamellae only. For example, students. One second, students. PS2 is found in grana lamellae. Yes. Here. You can see here this diagram right here. See here. PS2 is found where? Can all of you see the diagram, students? See here. This is PS2 right here. PS2. PS2 is found only on the grana. This is called as grana lamellae, no? Grana lamellae. Okay, grana lamellae only. Okay. While PS1 is found here also and here also. So PS1 is found on the stromal lamellae as well as the grana lamellae, on the grana also or thylakoid also. Okay. Thylakoid also, to remove the confusion. And we'll go back here. Here. So PS2 is found only in the thylakoid, while here photosystem 1 is found in both. Let's just say thylakoid for easy to remember. Thylakoid and stromal lamellae. In both the places we can find PS1. So all of you should know there is something called as photosystem. It is found everywhere. Okay, students, give me just 1 minute. Let me just show you if I have an image of photosystem. One second, students. I had it somewhere here. Students, this photosystem is actually made up of a lot of proteins and as such. Like this is how it looks like. This is how it looks like even electron microscope. Can you see, students, here? This is how it looks in electron microscope also. You can't really tell what is that. It's like bunch of lot of proteins and lot of electron carriers and light harvesting complexes. It is like this, somewhere like this. You can see here also, students. You can see here also. Okay, it is see. This is how the electron microscopy looks like. You can't really tell like how exactly it is, right? That is how it looks like. That is how it looks like. The entire photosystems. Photosystems. So remember, students, there are two types of photosystems present in the chloroplast. Present in the chloroplast. Now, students, now we come to the actual photosynthesis. Till now, whatever you learned is the basics to understand the light reaction. Because if you do not understand the basics right, this light reaction is going to be a nightmare for all of you. Okay. Now let's understand the basic, understand the advanced part now, students. Let's understand the advanced part right now. Okay. Now, first of all, students, the entire photosynthesis can be divided into two phases: light reaction, dark reaction. Light reaction is also called as light dependent reaction because it requires light for reaction to happen. Okay. Then there is something called as dark reaction. Dark reaction is also called as light independent reaction. Okay. And let me clear confusion right now, students. Small confusion. It doesn't mean light reaction happens in only daytime and dark reaction happens only in night time. Yes, light reaction happens in daytime because it requires light, but dark reaction can also happen in daytime because it doesn't mean it is happening in darkness. It is called as dark reaction because it does not require light. It is a misnomer. We call it in science. Okay, so this is called as dark reaction because it does not require light. That's all. It has nothing related to darkness. Nothing related to darkness. Okay, it's just that this reaction does not require light. Also called as light independent reaction. Now, students, what happens? It occurs within the membrane system of the chloroplast. So it happens in the chloroplast right now. Trapping of light energy by pigments. So what exactly is happening, students? In light dependent reaction or light reaction, pigments are trapping the light here. Pigments are trapping the light here, and with that, there is synthesis of ATP and NADPH. ATP, all of you know, adenosine triphosphate, and NADH is nothing but nicotinamide adenine dinucleotide. These are type of energy molecules. They are basically energy molecules. Okay. Now, students, now we come to the depth of light reaction. Now we come to the depth of light reaction. The depth of light reaction, there are many two types. There is something called as non-cyclic photophosphorylation. Then there is cyclic photophosphorylation. What exactly happening, students, here? This, as the name suggests, the first one is completely non-cyclic in nature, and also in this entire reaction, photophosphorylation is happening. The meaning is that light energy is used to make ATP. Light energy is used to make ATP here. That is what is happening here, students. Now, let me just draw this entire thing for all of you. So, if you are watching this lecture recorded, I would recommend all of you to draw with me. Okay, students. Now, what do we have first? What do we have first here, students? First of all, we have the chlorophyll A here. Chlorophyll A. First of all, students, we have the chlorophyll here. So, again, can anyone tell me chlorophyll A is going to be PS1 or PS2? Here we have chlorophyll A. First, we have PS21. PS2, which works at 680 nanometer. PS2 works at 680 nanometer, all of you know. Okay. Now, what is happening, students, here? What is happening here? Let me just draw this for you. Let's see, here we have the chlorophyll A. Okay. It has all the antenna molecules. Let me draw the antenna molecules also here. Okay, antenna molecules are here. Okay. Now what is happening, students? Here we have the solar radiation coming in. Solar radiation is coming in. Okay. Now what will happen, students? All of this light will be trapped by the antenna and also the chlorophyll A, and will be transferred to the reaction center. Remaining light is transferred to the reaction center here. See, all the light is being transferred here. So all the light trapped everything is sent to the main reaction center, that is the chlorophyll A. Chlorophyll A. Now, all of you know, students, in the previous I told you, when a pigment gets light, what happens? The electron gets excited here. The electron gets excited here. Okay, the electron gets excited here. Excited. Okay, electrons are getting excited right now. Electron getting excited. Now, students, these electrons are now taken up by primary electron acceptor. Primary electron acceptor. In this case, the primary electron acceptor is called as pheophytin. It is called as what, students? Pheophytin. It's called as what? Pheophytin. So pheophytin is actually picking up the electrons here. Pheophytin is actually picking the electrons. Now from here, students, there is a series of different electron carriers where the protein carriers where the electrons have been transferred here. Okay. Now, students, from here the electrons are sent to something called as PQCY-PC. 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 Okay. PQCY-PC. Done? Done, students. Now you're already wondering, students, what is PQ, what is CY, and what is PC? Obviously, it is very important doubt which needs to be addressed right now. Okay, let's address the doubt here. Once again, students. Students, if you understand this, the entire reaction is over here only. Okay. Now, let's address the doubt here, students. Let's address the doubt. Oh. One thing, students, what happened? Okay, one second. Where did it go? Okay, we have it here. Never mind, we have it here. Okay, just remember it is non-cyclic in nature. Non-cyclic. Photophosphorylation. Something happened, students. Okay. Now, let's just draw a box here. Okay, to explain all of you the simple full forms here, students. Okay, first one is called as PQ. That is nothing but plastoquinone. Plastoquinone. That is PQ here. Plastoquinone. Then we have CY. Cytochrome B6F complex. Cytochrome B6F complex, students. Okay, done. Then we have PC here. That is plastocyanin. PC is nothing but plastocyanin. So that is your PQCY-PC, how the electrons are being transferred here. How the electrons are being transferred. Electrons are being transferred here. Now, students, now where does the electrons go? Now the electrons are sent to the next partner here. The next photosystem here called as your chlorophyll B. Sent to the chlorophyll B. Just draw chlorophyll B, students, here. This right here, students, is a chlorophyll B here. This also has antennas. It also has antennas here. It also has antennas here. Now all of this light, again, here also there is solar radiation, students. Here also solar radiation is going to fall. It's going to fall here. Electrons coming from here also. They're coming from here. Now all this energy is again transferred to chlorophyll A. Chlorophyll A. Now, but in this case, students, this is called as PS1, which works at 700 nanometer. So this was your PS2, which is working at 680 nanometer. Here we have PS1, which is working at your 700 nanometer. So PS2 first, then we have PS1. Let me just draw a box around it so it will be easy to see also. So first we have PS2 here, PS2 here, then we have PS1 here, then we have PS1 right here. Okay, PS1 right here. Now what happens, students? From here also electrons were transferred. Then again here also we have primary electron acceptor. Here also we have something called as primary electron acceptor here. Now students, in this case the primary electron acceptor is called as iron sulfur centers. Okay, so here we have the iron sulfur centers here, FES, iron sulfur centers. Now how does the electron flow happen now? Electrons are now flowing again. Now again electrons are taken up by something called as FD, that is ferredoxin. Now after ferredoxin, students, electrons are now sent to the final electron acceptor. The final electron acceptor is none other than, students, none other than NAD plus. NAD plus. NAD plus, students. NAD plus. So when NAD plus takes the final electrons, it gets reduced. All of you remember, students, I told you. What is reduction? Reduction is nothing but gaining electrons or gaining protons. So in this case, students, in this case what is happening is this NAD plus is getting these electrons from here. It is getting these electrons. Along with the, students, along with that, it is also getting H plus from the stroma. Please remember this, H plus from the stroma. This actually is very, very important. This part is very important to explain chemiosmotic hypothesis. Very important. Okay. Now what is happening, students? This NAD plus is actually getting reduced now, gaining electrons, gaining protons, getting converted to NADPH or NADPH2. Now in this entire last step, there is an enzyme involved here called as FNR enzyme. FNR enzyme. Okay, FNR enzyme. Ferredoxin NADPH reductase enzyme. That will not ask you, student, generally. That is ferredoxin NADPH reductase enzyme. FNR. And that's how electrons are transferred finally back to NADPH, students. Finally, electrons are going to the NADPH. It's getting reduced here. Now, students, how is the electrons getting all the energy is getting continuously followed here? Like, there should be a continuous source for these electrons, right? See, pigment is there. As much as pigment is there, it needs a constant supply of these electrons also from here. See, enough electrons are supposed, enough are there, but it needs continuous supply also, no, of electrons to get excited here. Yes, it needs. So that what happens exactly, students, here in the inner side of inside the lumen part, towards the lumen part of the thylakoid. Let me just teach you that also, students. See, all these are high level concepts. Again, this, remember this structures here. This is called as a thylakoid. Thylakoid here. This thylakoid has an inner part. This inner part here. This inner part is called as lumen. Now, students, this entire reaction, non-cyclic photophosphorylation, it is happening actually somewhere here. It is actually happening somewhere here. Can you see here? Here we have, let's say, here I have the PS2. Here, let's say I have PS1. Then electrons are getting excited like this, somewhere here like this, they're happening. So this entire reaction is actually happening at this point right here on the membrane of thylakoid. Now, what is actually happening here, students? This is called lumen. Inner part is called as lumen. Now, in this lumen part, right, there is something called as splitting of water. What is happening, students? On the lumen side here, this is happening on the membrane, right? Happening on the membrane. On the lumen side, there is something called as splitting of water. Okay. Here, water is being split here. H2O is split into 2H plus O2 plus 2 electrons. O plus O plus electrons, plus 2 electrons. Now you might be wondering, sir, what happened to the rest of things? Students, understand very well. These 2 electrons are given here. These H2, right? These H2 are sent for ATP synthesis, which I'll teach you next concept. Sent for ATP synthesis. And this oxygen right here, students, this is the same oxygen which has been released during photosynthesis, which we breathe. Yes, this is where it comes from. This is where it comes from, the oxygen which we breathe. Okay, so that is the entire reaction of non-cyclic photophosphorylation. FD is ferredoxin, FES iron sulfur centers. So this is called as non-cyclic. With the help of this, there can be a continuous supply of electrons here. Continuous supply of electrons here. Okay. Now, students, let me just recap what all did we have here, students. In this entire reaction, in non-cyclic, did we see something called as O2 evolution or O2 release? Yes, there was oxygen was released. Okay. Now, second most important point, students, here, if you go back to this entire reaction, you might be wondering, students, why am I even doing all this, right? Why am I even doing this entire thing? Okay, student, this entire thing is done for the ATP production. For the ATP production, that is the main important thing, students. Between PQ and CY, using the electrons here, 1 ADP here is converted to 1 ATP here. Using the electrons here, students, and the H plus later on I'll tell you, 1 ADP is converted to 1 ATP. So 1 ATP formation is happening here. Simultaneously, right? ATP formation is happening here. Done? Done. Okay. So can I tell, in this entire reaction, okay, there is something called as ATP formation is happening. Yes. Then, student, there is NADPH formation. NADPH formation. Yes, it is happening. Then, students, in this entire reaction, there is splitting of water. Splitting of water is happening. Then what is happening, students? Then, students, can all of you see, students, this entire thing is non-cyclic. That is, electrons are transferred from here to here. So it is going in non-cyclic manner. It is going in a complete non-cyclic manner. The electrons are never coming back. They are never coming back. So this is called as non-cyclic photophosphorylation. It is completely, students, non-cyclic in nature. Non-cyclic in nature. It is completely non-cyclic in nature. And the last, the main point, this entire thing, students, if you see from an angle, doesn't it look like a Z here? Z? Yes, it does look like a Z. So this is also called as what? It is also called as Z scheme. It is also called as Z scheme. Okay, this is what you learn in higher classes. This is what you learn in the higher classes, the entire Z scheme. Crazy, students. Just have to imagine. This is actually happening in plants right now. If it's late, not now, but in the early morning, yes, it does. Okay. This is what happens in the case of plants, the entire non-cyclic. So if you know non-cyclic, cyclic becomes very easy. Cyclic is very easy right now. Very easy. Okay. In cyclic, we only have one photosystem. That is chlorophyll A, which has here PS1. PS1 works at 700 nanometer. All of you know that. Okay. Now here, this particular photosystem. Okay, photosystem. Right, one second, students. This particular photosystem also has antennas. It also has antennas. It also has antennas here. Whenever there is solar radiation, all the light is trapped by this and transferred to the main reaction center, chlorophyll A, which is in this case is P700. That is PS1 here, PS1. Now from here, electrons are transferred to the primary electron acceptor. Primary electron acceptor. That is nothing but FES again, iron sulfur centers. Okay. Now from here, electrons sent to FD. Now after FD, actually here, students, it doesn't end here. It doesn't end here. From FD, it actually goes to PQ, CY, PC again. PQ, CY, PC again. Okay. Now after reaching PC, the electrons again sent back to the chlorophyll. Okay. This is what actually happens in the case of your cyclic. Happens in the case of cyclic. Let me just write properly, students. Okay. FD, PQ, CY, PC. Now electrons sent back again here. Electrons are sent back here. Okay. Now between PQ and CY again here, between PQ and CY, here 1 ADP is converted to 1 ATP here. That is ATP formation is still happening here. ATP formation is still happening. Now again students, here between FES and FD, here also 1 ADP is converted to 1 ATP. That is your entire cyclic one. Now but the difference here students, in cyclic there is only ATP formation. There is no NADPH formation. There is no oxygen release also here. There is no splitting of water here. Okay. It's not called Z scheme. It is actually cyclic manner. So cyclic photophosphorylation. Done. Here are simple questions to all of you. See the difference you see students. Cyclic, what happens? Electrons move in a cyclic manner. Electrons in linear pattern, non-cyclic. See here students. Electrons go in a linear pattern. Okay. Now only PS1 is involved. Both PS1 PS2 both are involved here. All of you know. Non-cyclic both are involved. Here only one is involved. Electrons are first expelled from reaction center 1 here. Electrons are first expelled from the reaction center 2 here. See here. Electrons are first expelled from the PS1 here. Electrons are first expelled from PS2. Okay, from PS2. Now electrons return back to P700 after passing through ETC, ETS, electron transport chain. Right, electrons do not return back to 680 because it is non-cyclic in nature. Right, not return back. Synthesize ATP during the light reaction photosynthesis by cyclic passage, non-cyclic passage. Occurs in isolated chloroplast and photosynthetic bacteria. Occurs in higher plants, cyanobacteria. Occurs in unoxygenic photosynthesis because it doesn't require oxygen here. There is no oxygen release also here. Occurs in oxygenic. Okay, here. Photolysis occur here. Photolysis do not occur here. Photolysis do not occur here. Photolysis occur here. The splitting of water. Photolysis means splitting of water here. Now, final electron acceptor is P700. Final electron acceptor is NAD plus here. We can see here, students. Since it is cyclic in nature, the final electron acceptor is P700 again here. But in this case, the final electron acceptor is NAD plus here. Final electron acceptor. Okay. So that is your entire light reaction, students. That is your entire light reaction. End of it. End of the entire light reaction. Now, students, before I go right now, this I would like to teach in the next class because this is a much more you know what, I'll teach students, it's fine. It doesn't require much thinking. It is actually very easy to learn also. 10 minutes I'll teach it to you. Okay. 10 minutes, that's all. It sounds very difficult actually. Okay. Chemiosmotic hypothesis. It sounds difficult, but it is actually very, very, very easy. Chemiosmotic hypothesis. See, just try reading the definition here. Try reading the definition once. Okay. Synthesis of ATP due to facilitated diffusion of protons or H plus due to the proton gradient through the ATP synthase across the membrane leading to the formation of ATP. That is the complex definition of chemiosmotic hypothesis. Okay. But the simple definition is that, students, this entire process of chemiosmotic hypothesis, it explains how the ATP is formed. That's all. That's all is its theory. How does it form? That I'll teach you with the help of diagram right now. Okay. See, you saw here, right? In both cyclic ATP formation, in non-cyclic ATP formation, how this ATP formation is happening, that is what is explained by chemiosmotic hypothesis. That's all it is. Okay. Students here. Now, if you go back to here, students, this is what is happening here. Can you see, students? What exactly is this? All of you remember the thylakoids? All of you remember thylakoids? Tell me yes or no. Thylakoids. This right here is a zoomed out version. It is a zoomed out version of one thylakoid. This right here is one thylakoid right now. One thylakoid. Okay. Can all of you see it, students? There is your PS2, then there is PS1, PQ. Can you see, students? PQ, CY, PC. Here we have light is picked up by FES, FD, FNR, NAD plus, NADPH. Yes, light has been picked up here. Then picked up. Then you can see here in PS2 there is something called as splitting of water, oxidation of water, or photolysis of water is happening. So this right now here on the membrane, we can actually see something called as non-cyclic photophosphorylation is happening here in this membrane here. This inner part is called as lumen here. Lumen. So this is a particular section of a thylakoid. This is a thylakoid. This is one round circle. It is one round circle. Vowel actually. Vowel. Okay. Now, if for a chemiosmotic hypothesis to function, it requires four main things. Okay. First one, it needs an intact membrane. Then students, it needs a proton pump. Then students, it needs ATP synthase. Then it also needs something called as proton gradient. So here we have the intact membrane here. We have the proton pump here. Then we have ATP synthase here to make it. Proton pump is also here. ATP synthase. Now what about proton gradient? This we do not have. So this has to be created right now. Now meaning of gradient is difference. So right now the whole goal is to make a difference in proton. Difference between whom and who? Difference between the number of proton inside to outside. So you can see the diagram also, it says here high H plus in the inside lumen and low H plus on the outside here. So there is a gradient formation here. So inside the lumen we have high H plus, outside the in the stroma we have low H plus. Once this gradient is formed, then the chemiosmotic hypothesis can function. It does make sense, and ATP is formed. Now, how do we make this gradient? There are three ways to make the gradient. Okay. First one, listen to me, students. First one. Here, can all of you see, students, here? Because of oxidation of water, here, first point here is this H plus is forming here. All of you remember here. I told you if you go back here, students, I told you this H plus is sent for ATP synthesis. I told you here, this H plus here, it is sent here. So this is what is happening here. Everything is connected here. So first step is what is happening inside the lumen. H plus concentration is increasing because of the oxidation of water here. Now students, the second thing, this plastoquinone actually also work as a hydrogen donor. So it will use the electron energy and take one H plus in the stroma and bring it inside here. So again, H plus concentration is increasing inside. Can you see here? It's coming from here. This is H plus. Again increasing. Now when this is increasing outside, what can I say? If H plus is removed from here, H plus concentration is decreasing outside. That is the second thing here. This is second type. Now third way how the H plus is increasing. Understand, students. The third one is that in the last step here, students, what I tell you, for a conversion of this NADPH to NAD plus to NADPH, I told you one line here. If you go back here, students, I told you here, in this formation of NADPH, from the H plus is taken from the stroma. One H plus is taken from the stroma, I told you here. Yes or no? So can I tell right now, if you go back to this diagram, can I tell that because of this reaction, H plus concentration is decreasing in the stroma here again, decreasing. So outside it is decreasing, inside it is increasing. So with that way, there's a gradient formation is happening now. Now what happens? How the ATP is formed? Understand, students. Now inside what is there? More H plus. Now what happens is, students, these H plus will now start to pass through the ATP synthesis. Now every single time 4 H plus passes by, one ATP is formed. So more H plus inside, H plus is passed. One 4H plus will give you 1 ATP here. Every 4H plus will give you 1 ATP. You can see ATP is converted to ATP here. That's how ATP formation is happening. That's how ATP formation is happening in the case of where? In the case of your non-cyclic photophosphorylation as well as cyclic also. Okay, this is called as chemiosmotic hypothesis. So here I've given you the points also. You can read these points once. You can pause the video, read the points, and be happy, students. Be happy. And that is the end of today's class. That is the end of light reaction actually. So after this, I will teach you the advanced concepts of dark reaction. In a dark reaction, I will be teaching you something called as C2, C3, C4. And that will be the end of the chapter. Okay. So thank you so much, students. I'll see you in the next class. Until then, take care all of you. Hopefully you enjoyed the lecture. You understood the advanced concepts. Any doubt anywhere, let me know in the comment section. Because next time when I come live, I will take those doubts and address it. Okay, students. So bye-bye all of you. Take care and sayonara all of you. Bye-bye.